Good afternoon and good evening. I'm going to present results that we've been pushing forth for a couple of decades in terms of trying to understand the evolution of biological systems based on the information that we can gather from the genomic revolution. I want to thank Sohan and the organizers for providing me this opportunity to talk to this very diverse audience. So it's a pleasure to be here. The goal of our research for this many years has been back in time exploration, and the focus has been the study of the structure of molecules. That is the three-dimensional details of that structure, whether that information can provide us a window into function and evolution. The perspective has been always historical, meaning that we use the tools, the computational tools of phylogenetic analysis to pursue a top-down approach to the analysis of origins and to the history of the molecular components of life. So we've been generating chronologies. So going back to the presentation of Lowell Gustafson, the idea of big history, but this big history only encompasses the 3.8 billion years to the present. We've studied the chronologies of protein domains and loops, molecular functions, metabolism, metalomes and metal utilization, biocatalytic mechanisms, folding, molecular clock, defining a molecular clock of folds, studying the history of transfer RNA, RNase P, RNA, 5S, ribosomal RNA, and of course of the ribosome, and many others that I've not listed here. But the idea behind all this effort has been to search for a generative process that is ultimately responsive for biological complexity. And of course, in order to do that, you need to have actual data that would support an hypothesis of such a magnitude. So I want to start by introducing, of course, somebody that you may know, Herbert Simon. He was a Nobel laureate in economy that in 1962 put forth this very insightful paper where he defines systems, and he basically founds, with many others, the system, complex system theory. The idea that systems are structured by the interrelationship of their parts. So basically, parts make up wholes, and this is what needs to be modeled. And the recognition that organized entities that are structured in no simple ways. So this is the very common idea of the whole is more than the sum of the parts, isn't it? That we need to somehow understand this complexity in some way. One aspect that he also introduced at that time was the idea that systems are hierarchical. Of course, he did this, illustrate this with the idea of how a company, for instance, is structured. But at that time, remember, this is 1962, he also used proteins and nucleic acids to somehow put forth this proposal. The idea that hierarchy, an organization that is ranked according to some authority, is somehow responsible for that structuring of the biological system through levels of organization, through nesting, through balance of that nesting, and through authorities. You know, authority meaning that there are some levels that control others in this complexity. So one interesting thing that has occurred during the past 2 decades is the realization that mathematically we can model a system of parts and wholes with a mathematical contraction, which is the network. In this network, we define nodes as parts of a growing systems, and the connections between those nodes as the interrelationship of the parts that make this system more cohesive or stable. So this is basically the entire idea behind biological systems. So now what I'm going to introduce is the idea that biological systems grow. And we know we have plenty of evidence of this. I'm just going to illustrate this with 4 cases. One is the evolving Tyrannosaurus family. A recent paper has shown that the size of these animals increase in evolution. So you have an incredibly complex organism increasing in size as evolution proceeds. We have the same situation with viruses. We can build a periodic table of capsid size and complexity, and there we can showcase how viruses are also growing their systems for particular purposes. The same we can say about biodiversity. There's plenty of evidence, paleontological in particular, that supports the idea that biodiversity increases with time, in this case in land and sea. Well, let's go molecular. The ribosome also increases in size in higher organisms. Here you have a measurement of the length of the large subunit of the ribosomal RNA and how it increases considerably in higher organisms, as you can see by the size of the circles there that measure that length in nucleotides. So there's no doubt that that systems grow and that in the process they accrete parts to make those systems bigger. So this is the idea of accretion, a process of growth and increase, typically by the accumulation of entities and their interconnections. Now, the interesting thing is that we can think of networks also accreting, the process of growth and increase of nodes and links defining a network system, including this associated properties. So this is simply a modeling device of how we can visualize a biological network with a network structure, a biological system with a network structure. So it is clear that accretion appears universal. You know, we see accretion occurs in galaxies and starts in planets and molecules. We can see it in cities, and I'm going to showcase 2 cases of, you know, the growth of, for instance, the high-rise buildings in Lower Manhattan, where you can see this start in the late 1800s of growth because of growth of population. At the bottom, you see the planetary accretion of cities in the course of thousands of years. It's an irreversible process where, you know, the number of these constructs that come from human activity increases slowly in time. And at some point in the description in the bottom, you're going to see a big bang occurring of massive construction of cities throughout our planet. So this is not new. This idea that there is that we can extract interesting information through generating big history at different levels of complexity. So here I showcase with this diagram, well, how accretion occurs at different levels. It occurs at planetary level, of course, but it also manifests at cellular level. So we can put forth a cellular history. We can put forth a protein history, and this is where we've been studying and focusing a lot our efforts. And of course, we can even construct the history of a complex that is crucial for cellular life, which is the ribosome, responsible for protein synthesis. And so we can basically align all these chronologies and attempt to make a global view of this complexity. So, but this requires a theory. And Jay Mittenthal, my son, and I in 2012 put forth a model. a model that would describe how all this complexity would unfold. And we call this model a biphasic model. It's a bow tie model, the idea that the evolution of biological structure is driven by a double tail of accretion and change. So accretion brings together disparate parts to form bigger wholes, like for instance, forming a larger ribosome. And then change provides opportunity for growth and innovation. And it's biphasic because it has those two elements coupled together. In a first phase, parts are first weakly linked and associate variously. As parts diversify, they compete with each other and then are selected for performance. And the emergent interactions then constrain the structure and organization of that emergence. And then this causes parts to self-organize into modules with tight linkage. So here I define the word module, which is highly used in evolutionary thinking, the idea that of components of that system that interact more with themselves than with the rest of the system. Well, then after that first phase, variants of the modules evolve and become new parts for a new generative cycle of higher level of organization. So this is an attempt to somehow describe why we have complex systems and why we have hierarchical systems. And this is what I'm going to be unfolding in this talk. This model has predictive power. The double tail explains, for instance, the emergence of nucleic acids and proteins as successive cycles of module creation. For instance, in the case of nucleotides, they basically assemble into small stem structures that then they become more complex and more stabilized by, for instance, stacking interactions. And then these stems associate with other stems as the accretion continues. And then later on, you will find the formation of junctions, which are these elements that really provide increased stability and structure to an RNA molecule. And of course, this continues all the way to form modern RNA and complexes. The same we can think of proteins. Therefore, starting with small amino acids and the peptides, building loop structures that have only secondary structure organization. And then these loops assemble into complex three-dimensional structures, domains, and these domains then assemble in proteins as multi-domain complexes, and then these form modern proteins and modern complexes in modern biology. So is there support for this kind of view? And I contend that there is, and that the genomic revolution and structural biology has provided us a lot of information to make this a reality and support this theoretical construct. The double tail is supported by universal ancient cores and derived phylogenies. This idea that given an origin, an accretion occurring, then we can unfold a phase one that provides this universal core. And then once the core is constructed, then the elements of the core start diversifying and providing a periphery of diversification. And so these Venn diagrams that I'm showing here are genomic distributions of a number of things that we've been counting, in syntheses in the past several decades. Protein loops, structural domains in proteins, gene ontology terms of molecular functions, RNA families, for instance, in the RFAM database, ribosomal proteins, enzymes, structural domains that exist in viruses, or the variant types, the shape of the virions that are responsible for causing disease like COVID-19. So, and as you can see, in all cases, there is a core in red there with those numbers that is universal. And then you have all this incredible diversity unfolding around it as they unfold in the three superkingdoms of life: archaea, bacteria, and eukarya. So this already supports this idea, but of course, this is comparative genomic approach. This is not a phylogenomic approach. We are not retrodicting back in time. So we need more information, we need more support for this, this idea. So the double tail, when we proposed it, was already supported by considerable evidence, the existence of a developmental hourglass, diversification of macromolecules and protein structures and shapes of RNA molecules. There was evidence supporting this, the diversification of networks, early metabolism, evolution of codes, evolution of development and epigenetics, all arguments that we use to support this double tail. We also reconstructed molecular history. We had the data at that time, and we had chronology in billions of years of how, for instance, metabolism unfolded. And there you can see already how the number of structural domains that were appearing in relative time at the bottom was generating this biphasic pattern. You can see that clearly in this plots of the different structural domains in enzymes that were appearing at different times that are here binned. So, so this is kind of a chronology provides evidence to support this biphasic tail. So let me explain exactly how we build chronologies first very quickly, and then showcase how this can be translated into a study of network structure. What we've been doing for a long time, we've been permanently surveying genomes, genome information, and now we have over 100,000 sequenced or in the process of completion of sequencing, and many of those highly curated. We then mine these genomes with machine learning methodologies to assign protein structure to the encoded proteins. And then we generate demography tables. These demography tables here are then subjected to normalization, encoding, and phylogenetic analysis. And we produce through the phylogenetic analysis trees of domains. These are tree that describe the history, domain history, for instance. And we can also construct trees of proteomes. And these are trees that describe the tree of life through the information in a proteome. In the same way, we can study RNA structures, and we can infer that structures directly from sequence through alignments and through many other approaches. And we can construct, by the same token, a tree of parts and a tree of wholes, a tree of entire molecules or a tree of substructures of those molecules. In both cases, the case of trees of substructures and trees of domains, they provide a way to understand how a molecule or a collective of molecules like a proteome has increased in size as evolution has proceeded from the origin of the first protein domain to the present, where we have thousands of them making up the protein complexity. So, and we can do this at different levels of molecular structure. There are classifications that people have been gold standards of structural classification in proteins. And one of them is, for instance, the CAF classification of proteins. The higher hierarchical level of classification is what is called the architecture level. And there's a restricted number of those architectures that define the world of proteins. And this tree basically tells the story. Here at the base of the tree, you see bundles of alpha helical elements and sandwich structures of helices and strands, which are the most ancient, and they are associated usually with membranes. And then later on, at the most derived aspects of branches of this tree, you see structures are much more complicated. Here on the top, you can see a three-bladed propeller, which is an incredibly complex and convoluted structure that is highly symmetrical, that exists only in eukaryotes. So there is a progression, and this progression provides us in time a lot of information. And well, what we can actually do is take all that information and translate it into individual aspects of that molecular complexity. For instance, we can study the evolution of ATP synthase complexes, and we can basically map the ages of appearance of the individual domains so that we can have a chronology of how this complicated structure that is fundamental for managing the energetics of the cells has grown in evolution, has at the beginning basically interacted with the membrane, but then has added a central shaft that is fundamental for the rotation of the it's basically a motor. It's a motor, a molecular motor that generates energy, and as you can see, this has taken 3.8 billion years to produce this complex. And we know this because we have a molecular clock of folds that allows to relate time, relative time from our chronologies, from our phylogenomic analysis, to basically time from a geological perspective in billions of years. So therefore, we understand that this complex has taken a lot of time to perfect, and in doing so has added accreted elements and added complexity to its makeup. So the same we've done with the ribosome. We basically trace the history of both RNA and proteins, and we studied how coevolutionary interactions were occurring as the complex started at about 3.3 billion years ago to accrete component parts, both of our ribosomal RNA and ribosomal proteins. And then at 3.1 gigayears after, during this initial phase of accretion, it generates a processivity core where everything comes together and is able to basically produce a prosynthetic function. And this core continues to evolve up to 1.3 billion years ago. At that time, you will have the exit tunnel complete, therefore allowing to police the folding of the proteins in a co-translational way. And then after that moment, then we can start seeing this second phase where the core, the ribosomal core, starts to unfold further accretion specific to the different branches of the tree of life. Specifically in humans, for instance, we have a crystallographic entry describing the human ribosome, and has very little to do, except for that core, with the ribosomes present in prokaryotes, archaea and bacteria. So there is a clear biphasic double-tail pattern evident in these accretions when you start mapping history onto molecules. So now what happens is that we can turn the biphasic tail into a double tail of network creation or network structure creation. So we can postulate that the double tail predicts the evolutionary emergence of network structure that is both hierarchical and modular. So in a first phase, the emergence of hierarchical modularity gives rise to small, highly connected subnetworks that you can see in color there in the middle and with module emergence. And then in that second phase, those modules coalesce by combination into higher modules of network structure, all of which enhance the cohesiveness and stability of that network structure. So we can, with a network paradigm, we can therefore put to the test the idea whether the model matches reality. And for that, we need to reveal that hierarchical modularity increases in the course of evolution. So we've done this by studying recruitment of protein enzymes in metabolic pathways as time proceeds. or recruitment of elementary functional loops that are central for the functionality of proteins into structural of domains of proteins. There you have an example of how loop EFL8 makes the central piece of the functionality of the active site of Rubisco catalytic domain. Or for instance, making a recruitment of structural domains into multi-domain proteins, where individual domains come together to form vast numbers of multi-domains proteins that populate the protein world. So we've done this, and I want to showcase very quickly, because I'm getting close to the end, of the evolution of metabolism in the top there, and the evolution of domain organization at the bottom. We measured modularity with a number of algorithmic implementations that study communities of nodes connected in networks, the Louvain, the Newman-given methodology, the fast-greedy community algorithm, and other different systems. And in all cases, we've seen hierarchical modularity unfolding as time progresses in the network. So that validates our methodology. We've done this with metabolism, and this is a database that we have. It's called Molecular Ancestry Network, MANET. And what it does is basically colors with different ages the age of the individual structural domains that make up the enzyme activity in metabolism. And we've done this for the entire metabolic networks. So we have, we use the Kyoto Encyclopedia of Genes and Genomes definition of metabolism, and which defines metabolism as a hierarchical level of mesonetworks, subnetworks, and enzymes. So we are mapping enzymes to subnetworks. In this case, for instance, we have pyrimidine metabolism subnetwork, but there are more than 100 more than defined the complexity of metabolism. So we have, we can actually make bipartite networks where we have subnetworks on one side and enzymes on another as being nodes, and the connections are things that are shared, enzymes that are shared by the individual subnetworks. So when we do this, and study how the metabolic network has grown from its origin to the present, we see there an increase of that Louvain measurement, the Q value that you see there unfold in red and orange, increase in time. So hierarchy and modularity increases. That's what Louvain measures. But if we measure the clustering coefficient of the network, which is a measurement of community structure, and we plot that against the number of connections that a network holds, and if we see a power law relationship unfold, then we can say that that modularity is coupled to hierarchy. And exactly that's what we are finding here. We find that the power law scaling of the clustering coefficient with the number of links increases in evolution. And not only that, that there is a finer granularity occurring at a lower level of hierarchy when we are considering enzymes and not subnetworks. As you can see, this is what basically Herbert Simon predicted in 1997 when in his models of bounded rationality, where he said each of the parts of a nearly decomposable system has strong internal links among its subparts, but the several top-level parts are bound together with each other only by comparatively weak linkages. So this is the idea that the more granular the world becomes, the more tightly bound things are together, and the more hierarchical and modular that world becomes. So we've done the same thing at the combination of domains in protein level. And this is a network unfolding in time from top to bottom. And in the center, you have the 5 different kinds of definitions of that network that relate protein domains to each other, whether they are able to combine in a protein exist in a particular organism. And of course, here we study thousands of organisms. So it's not only one organism. We use the range of diversity throughout the protein world. And we find out that there is a Big Bang. We've described this already in 2009, the existence of a Big Bang in the evolution of protein structure. But you can see it unfold there. You know, you can see the complex networks appear at the bottom. To showcase this in a different way, here we have the actual time proceeding from back to the present. And as you can see at the beginning, very few links, very few protein domains were combining with each other, you know, but that changed. And at some point, some particular domains were very important donors, became important donors to building complexity in protein structure. And then at some point you have the Big Bang occurring. You can see those dots in the middle. Those dots are basically domains that are highly important in terms of donating their domain to the formation of complex multi-domain structures. So this is telling you that the biphasic structure of is unfolding at the network level, and that hierarchical modularity, which is the outcome of that biphasic process, unfolds in evolution. So to my surprise, some years ago, I discovered the Strasbourg papyrus. It's the oldest pre-Socratic thinking text that was embedded in a papyrus. It's the only one. It's one of the few that exist, but this is the oldest of them. It was decoded in 1984, but Richard Janko, which I've been collaborating for the past 2 years, which is a curator in the University of Michigan, and he has been helping me kind of go through the papyrus and study this double tail that the papyrus proposes. The text starts in the following way: a double tail I'll tell, at one time one thing grew to be just one from many, at another many grew from one to be apart. And this is the biphasic theory, isn't it? The module creation theory. In a study of this ancient text, I found Darwinian and network thinking 2,500 years ago, and a text that anticipates this biphasic theory, but it also proposes the existence of fossils. It proposes a system of classification of life. It's incredibly powerful, and for me very challenging, because that implies that the ancient Greek had already incredible insight into our natural world. So what are the messages? As I close, take-home messages. Hierarchical modularity increases in network evolution. The world becomes more hierarchical and granular. Evolutionary constraints of network structure are stronger at lower levels of biological organization. And the history of network structure manifests at many levels. I have not shown nanosecond data, but we've been also studying conformation of molecules, and they show also a biphasic pattern of module creation. So I end here with perhaps this very interesting video from Peter Gabriel, Claymation, which talks about big time and a big history, basically. It talks about big history and the process of accretion. So thank you very much for your attention. I welcome any questions you may have. So
thank you.